All right, so probably another minute, but yeah, thanks again, Ethan. Before we properly get started, it's a really stressful time at the moment with grant deadlines approaching
Yeah.
for NHNLRC and ARC. So we really appreciate the time you've taken to prepare your talk and turn up today. I'm certainly looking forward to the exciting science and having a break from my own little world and documents. And I hope that everybody in the audience is as well.
Great. Yeah, it's my pleasure. No problem at all. It's a nice break as it hopefully provides an hour-long break for some of you to think about something different for a little while. It's been fun to refocus on this work. And as I'll say in the introduction, it's a little bit of a different talk for this audience, and that's been fun as well. So.
Yeah.
Okay, so if we just hold for a moment there, Ethan, sorry, there was a slight problem with setting this up. There are some people over in the meeting. I'm just telling them to get across into the webinar.
Sure, yeah.
Hopefully they will be here in just a moment with us.
Yep, all good.
Okay, Steve, I'll probably kick off soon anyway, just with an introduction, and then they won't be missing too much.
Yeah, sorry, just making sure everybody's got the details to get across here.
Okay.
Some slight confusion there. Okay, I think we're good to go.
All right. Well, welcome everybody to the Monash Advanced Microscopy webinar. My name is Alex Coombs. I'm a group leader at the Monash Biomedicine Discovery Institute, and it's my pleasure today to introduce our speaker, Professor Ethan Scott, who joins us from the Queensland Brain Institute at the University of Queensland in Brisbane, Australia. Professor Scott completed his undergraduate studies in molecular genetics at the University of North Carolina in Chapel Hill. He then moved to Stanford University for a PhD in developmental neuroscience in the Drosophila model organism, and then joined Professor Herwig Bayer's group at UC San Francisco, where he developed the GAL4 enhancer trapping system in zebrafish, building reagents for the exploration of neural networks that his lab continues to work on today. Professor Scott his research blends optical physics, neuroscience, and computational biology to describe sensory networks in zebrafish. And today we're going to hear about their development and application of custom light sheet microscopy for calcium imaging in vivo. It's really exciting. Ethan gives a great talk. And if anybody has any questions, please post them in the chat as we go, and we'll come back to them later. So thanks very much, Ethan. Take it away.
Yeah, thank you, Alex. Thanks for the introduction. Thanks very much to you and the rest of the organizers for the invitation. I'm really thrilled to be talking to this audience. I'm not a physicist by training, but we have physics that's been going on in the lab for quite some time now, and it's grown to be a big part of my interest right alongside the biology and the computational neuroscience. For this audience, I'm particularly excited. I'm going to slant the talk a little bit differently from what I normally do. So I'm going to give more technical detail on the microscopes that we build and why we build them the way we do. I'm going to give a little bit on the analyses we do with the data that we get out. And I'm going to minimize my talking about the biological and the sensory neuroscience stories. Again, to highlight the microscopy, I will tell one biology story, but that's one with sort of a fun physics slant to it. And I'll wrap up by talking about some of the further advances we're working within these technologies, the reasons why these advances will be helpful to us as neuroscientists, and then with some very fresh, unpublished data, some of the problems we're running into. So you may have questions about that, and certainly if any of you have comments on that with your perspectives on the microscopy and on the physics, I'd be all ears. That would be fantastic. So thanks for coming. I'll get into it. As a neuroscientist, I think everybody in my group, I think indeed all neuroscientists, whether they know it or not, are all studying the same question. They're all studying how it is that stimuli in our surroundings can go through this vast and complex network that is our brain and produce appropriate adaptive responses from what the world presents it with. So whether they're studying what goes wrong when this system is awry, whether they're studying how this system develops in the first place, or whether they're studying, as we do, how information flows through this system to move from the perception of a stimulus to the delivery of a response, we're all interested in some aspect of this question. Historically, in order to observe or to do experiments on the flow of information through the brain, there have been a couple of broad approaches. One, exemplified by fMRI, allows you to observe activity across the entire brain. But these technologies lack cellular resolution. So you're dealing with voxels that themselves may contain hundreds or thousands of individual neurons. If you're interested in the activity of, or you'd like to manipulate the activity of individual neurons, for the better part of a century now, there's been electrophysiology, which has extraordinary sensitivity, extraordinary time resolution for individual neurons. You can see exactly what's going on with the physiology of individual neurons, and more recently, tens or dozens or now many hundreds of neurons, but still far short of a comprehensive sampling of what all the neurons in the complex brain are doing. And the consequence has been that between these whole brain methods that don't resolve individual neurons and these methods for imaging individual or small number of neurons, we miss a critical couple of scales, those of local circuits and the connected neurons within brain regions that process information, and indeed of brain-wide networks. The brain is little more or less than a vast network of neurons that are interconnected in a way that provides advantageous information processing for its owner. So this gap at the level of circuits and networks has been a major obstacle to understanding the brain, and one that has been at least theoretically addressable with the advent of a set of technologies that I think this audience will be quite familiar with, optogenetics. So just at the risk of, just to avoid the risk of leaving anybody behind, I'll repeat probably something that most of you will be familiar with. Optogenetics, these are proteins that respond to light and can be used to detect or manipulate neural physiology. Opto, of course, light. And genetics captures an important property of these tools. They're genetically encoded, they're proteins. And so that means that if you have the genetic means of expressing them specifically in cells that you're interested in, you can target these tools to specific cell types, or if you so choose, you can express them pan-neuronally. The great advantage to this is it's sort of in parallel with brain-wide approaches, only not being able to resolve individual neurons when imaging. Approaches for manipulating the function of the nervous system as a whole, transcranial magnetic stimulation or even surgical ablations, don't resolve individual neurons, don't allow you to address circuitry. EFIS, which allows you to manipulate the activity of one or a few neurons, it doesn't allow you to manipulate the whole package. So in the same way that there's an obstacle to observing circuits and networks, there's an obstacle to manipulating circuits and networks. And this is where optogenetics still holds the potential to be enormously effective because it provides broad yet specific manipulations of neural activity. Something I'll talk more about today is its potential then for brain-wide imaging of neural activity. If instead of putting, say, a channel or a halorhodopsin in order to manipulate physiology, you simply put in a fluorescent indicator of neural activity, a calcium indicator in the case that I'll talk about today. So these technologies have been around for about 15 years. They were very splashy when they came out. They're working their way into a lot of what neuroscientists do, but I think they still have yet to fulfill their potential really to break down circuits in important ways. And that's part of what we're trying to work toward. Now, in order to do optogenetics in vivo, you need to get light into and out of the brain. And this is a very famous picture from the early days of optogenetics from Carl Deisseroth's lab at Stanford of a rat. And if you want to get light into the brain of what I would call a big, furry, opaque animal, you need to take an approach like this one, where you drill through the skull, have a cannula, and then insert an optical fiber into the brain in order to deliver light locally within the brain. And that's rather low throughput. It's difficult to make it perfectly consistent from animal to animal. And in a brain this size, even this just illuminates a portion of the brain, which depending on what you're trying to do can be an advantage or a disadvantage. The approach we've taken instead is to move to an animal system that is much more amenable to getting light into and out of, and that's the zebrafish. So zebrafish larvae have two inherent biological properties. Zebrafish didn't evolve for optogenetics, but if they had, they wouldn't look much different. The properties are that, like most fish, zebrafish have external fertilization. So the sperm fertilizes the egg in the open water. You can observe that process, and you can observe the entirety of development from that time forward. An example is here, where you can see an animal. This is a one-day-old zebrafish larva or an embryo. Its eye is there. This is the yolk sac. This is where it's deriving its nutrition. And this is the developing long axis of the animal. We're going to watch from 1 day old until 4 days old, simply as this animal develops in the water. And simultaneously, we can observe the development, in this case, of the circulatory system. This animal is expressing a fluorescent protein in its developing circulatory system. And so in contrast to mammals that have critical developmental processes occurring while the animals are still in utero, you can watch this process play out just under a microscope. You don't need to dissect, you don't need to interfere with the development in order to observe it. And importantly, in addition to being external, they're optically transparent at early life stages. So you don't just observe the surface of the animal, but particularly in dark field applications, you can look at targeted structures developing. So I showed you that for the circulatory system. Here's a particularly beautiful example from the nervous system, where we'll watch a video of the developing motor neurons down the spinal cord of a zebrafish. And you can see they're reaching their axons out, finding the targets among the muscles, innervating those muscles. There are no particular tricks here aside from producing the fluorescence signal. This is a normally developing healthy animal. After this experiment, it could go on to live a long, fruitful life in the way that some fish do. So these are extremely amenable to getting light in and out. And that has advantages both for observing activity, as I'll mostly talk about today, and also using optogenetics to manipulate activity. So what are our approaches for doing that? And I think this will be very familiar to most of you, but I'm just going to go over it very quickly, especially the light sheet microscopy, which will be the topic of the next several slides to follow. Our fluorescence signal and the way that we observe activity comes from GCamp. Mostly I'll be talking about GCamp 6S in this talk, but we're moving on to the GCamp 7 and the GCamp 8 variants in the lab. This is a fluorescent protein. It's fundamentally a GFP, but it's fused to a calmodulin element that when bound to calcium leads to a brightly fluorescent protein, and when not bound to calcium leads to much, much weaker fluorescence. And so if you have a population of hundreds or thousands of individual GCamp proteins within a cell, and if that cell has a wave of calcium, when it's active, you'll get a blink of fluorescence under the microscope. So to make a long story short, if you have a means of looking at the whole brain and a means of detecting these blinking points in space, you have a means of detecting activity in individual neurons across the brain. Now, the microscopy we use to do that is spin microscopy, selective plane illumination microscopy, or a lot of people just call it light sheet microscopy. And the way that this works is you take a collimated laser beam, and then using a cylindrical lens or using galvo mirrors, you sculpt this collimated beam into a sheet. And you place your sample right at the narrowest part of that sheet. That leads to the illumination of a plane of that specimen. And then orthogonal to that and parfocal with that, you have a detection objective, an imaging objective that captures the green fluorescent light coming out of here, brighter in cells that are active than in those that are not. That goes up to a camera. And the advantage of doing this is that with a simple CMOS camera, you can then, rather than scanning, which is a fairly slow process, you can take a snapshot of an entire plane at a time. And if you have a means of doing it, a desire to do it, you can then scan through and do volumetric imaging by looking at different depths within your specimen. So this is the approach we're taking. It's an approach a lot of people use, but very often using off-the-shelf light sheet microscopes. For reasons that I'll highlight, we've always built our own. And for this audience, I'm going to spend the next several slides talking about the design of our microscope and the rationale for those designs, and a couple of the design details that have worked well for us. So in order to get a zebrafish larva under the microscope, we have a 3D printed chamber. We have a lot of different forms of it, but they have in common that they have a stage. You put a larva here embedded in agarose on the stage. It's fully embedded for some applications. Sometimes we want to be able to observe tail movements in this animal. And when that's the case, we cut the agarose away from its tail so it's free to move. So the animal can move its tail while we're doing the imaging. We then put cover slip walls onto this chamber to create a fluid-filled chamber like the one you can see here. And we have illumination objectives here. These are the objectives that are producing the light sheets. Whenever possible, we use pairs of light sheets that overlap from different directions because it gives a more uniform and a more stable illumination field. And then here's our imaging objective that is focusing on the animal within the same plane that's being illuminated. So one of the problems that we had in this system, and it's particularly acute in intact animals and in vivo, is that we were getting striping when we did this. And so that effect, I'm gonna give just step away for a second and talk about a little optical physics anecdote, and then I'll return to the main talk. A problem that you get with light sheet microscopy is if you create a sheet of light where all the rays are parallel, and if there's some sort of an optical obstacle in the specimen or upstream of the specimen, you get a dark patch, you get a streak, a stripe within the animal. And in this region, you either don't get any fluorescence imaging, or worse still, there are cases in which this flickers, and that throws out your detection of neural signals terribly. So Michael Taylor, a postdoc in the lab at the time, who now has his own lab down at the University of Otago, introduced a 1D diffuser, or a line diffuser, as they're called, to the light sheet. So the light sheets that he was producing, rather than having parallel rays, were having diffuse rays. Now, to be clear, the sheet was still a sheet, but instead of being a sheet where all the rays were in parallel, it was still a sheet. It was still hopefully just a two-dimensional structure, but where the rays came in at various different angles. And what that means was that if you got an obstacle, rather than having a persistent stripe downstream of that obstacle, the sheet would quickly repair. It would heal itself on the far side of that obstacle. The nice thing about this is it's very cheap and easy. And so here, in a case without the 1D diffuser, you're using galvo mirrors to produce a sheet of light with parallel rays here and subject to striping. Here, once it goes through a line diffuser, you get this distribution of angles across. You get a meshwork within the light sheet, and that's then more uniform. This is about a $150 element. You just drop it into the light path, and to a large degree, that solves the striping issues. You can see here the same animal imaged on the same microscope with and without a line diffuser, and you can see these deep stripes in the animal without the line diffuser. You can see those are largely alleviated when the 1D diffuser is in place. If we look in closely, remembering that our goal here is to identify and extract signals from individual neurons, we always have to go through and segment out in an automated fashion where the neurons are. And when you have a stripe like this, that's impossible to do. So in a best case scenario, we lose data when we get stripes like this. And in a worst case scenario, we get spurious data from these points. We get artifacts that interfere with our interpretations. Again, those problems are largely alleviated with the 1D diffuser. So that's the approach that we take to build the light sheet. Let's talk a little bit more about the imaging column and the way that we built that here. This work largely having been done by Idia Favre-Boule. She was a PhD student, now a postdoc in the lab, and a great deal of this talk, especially this sort of microscopy and physics-oriented talk, she's a physicist, and she did most of what you'll see today. If you move up the imaging column from this detection objective here, something we always do is we include 1 or 2 or 3 empty filter cubes, first thing out of this objective. And this provides us with a lot of leeway. For instance, we can image a different wavelength. We can put a dichroic in here and another camera and image something that's happening in a different wavelength. For instance, if we want to image movements of the animal's eyes separate from the calcium imaging, we can take a certain bandwidth out of there, project that onto a camera, and view the eyes that way. It also gives us the ability to bounce light down into the specimen using an appropriate objective that's already in place. So I'll talk about examples of using that for optical trapping and also for the delivery of sculpted holographic light into the brain near the end of the presentation. That's why we have these empty filter cubes here. And then as you move up the imaging column from there, something that will stand out is this is a very tall column all the way up to the camera at the top. It's about 1.5 meters tall. And that's strange. So why did we do that? And that relates to the fact that we want to do volumetric imaging, and also to the fact that we want to minimize off-target sensory events in the animal's chamber. So we're looking to deliver, say, very subtle acoustic stimuli to these animals. We don't want a lot of things going on in the chamber while we're producing those very sensitive, those very subtle stimuli. If you wanted to do volumetric imaging in a specimen and you have a light sheet, you have a specimen and an objective, the simplest thing to do is simply to move your specimen through the light sheet and leave the objective where it is, leave the light sheet where it is, and you can look at different depths that way. But we didn't want to do that because that would involve a piezo drive that would be creating noise. That would involve the creation of vestibular stimuli, of acceleration for the animal, and who knows what processing we might see off target as a result of that. The other approach most people take if they want to do volumetric imaging is to take the brain and, using usually galvo mirrors, scan the sheet through the brain without moving the brain, and then have a piezo driver on the objective that keeps the appropriate plane in focus. So you're coordinating movements of the objective and the light sheet, and you're looking at different depths that way. But again, the idea of having a piezo driver here on the objective is going to create sound, it's going to create water flow, it may create visual aberrations for the animals, and we don't want those things going on while we're imaging sensory activity in the brain. So the solution to this conundrum that Idia came up with was, yes, to hold the animal still and move the light sheet through the animal, but rather than moving the imaging objective, she inserted an electrically tunable lens here in the imaging column. So this is a lens that changes shape as you change the current that's put across it. And as it changes shape, the depth within the animal that is in focus on the camera's detector changes. So you have control over where in the brain you're imaging without moving anything within this area here. So everything here is still, the light sheet scans through, and the electrically tunable lens controls the depth. You coordinate the current across the lens with the movements of the sheets in order to do volumetric imaging. If you're curious, the reason it's so long is that there's a linear relationship between the length of this tube and the depth that we can focus across in the brain of the animal. So we let the zebrafish brain dictate that, and Idia kept building the column longer and longer, and we kept getting greater and greater capacity until we hit what we needed to image from the dorsal to the ventral surface of the brain. It's about 300 microns, and with the particular optics we're using, that led to about a 1.5 meter long imaging column. So this is our strange and sometimes beautiful beast that we use. And hopefully I've explained why we've designed it that way for the particular applications we have. We have two of these. They have slightly different optical properties. They have slightly different capacities in terms of being able to do, for instance, holographic optogenetics versus simply calcium imaging. And as I'll say at the end, we're upgrading one of them to 2-photon light sheet right now. So they perform slightly different tasks, but largely overlapping. And really the core of the instruments are the same. So using that approach, we can collect data like these. This is a larval zebrafish. It's 6 days old. Using the electrically tunable lens and moving the planes, we can focus from now the dorsal surface down to the ventral surface of this animal. And you can see it's pretty easy to resolve individual cells. This is a GCamp line where GCamp is restricted to the nuclei of neurons, and that makes them easier to segment later on. So as we move through, we collect images like this. This is an animal receiving an auditory stimulus, an acoustic stimulus. You can see auditory responses in various parts of the brain right here. And the approach that we take, because we want to be able to gauge interactions across the whole brain to the same trials, is we scan through the brain. Typically, our experiments involve taking 50 different planes in Z, 100 frames per second. So that's 2 volumes per second. So while you're watching one movie that we're collecting right here, we're simultaneously collecting another 49 movies. We combine all of that into an approximately volumetric representation of the brain. The step size at about 5 microns is very similar to the diameter of these neurons. And so while we're not systematically imaging every neuron, we're coming pretty close to that. And we're certainly deeply sampling from a representative group of neurons across the brain. So what do we do with these data once we have them? The first thing we need to do is extract signals from individual neurons. So we do a certain amount of pre-processing. This is largely to eliminate the effects of drift over the course of, say, a 30-minute experiment. And to illustrate this, I'm going to talk about one structure, the optic tectum. I'm going to talk about a single plane in a single structure on a single side of a single fish. This will be about 400 neurons in the examples I showed, but many of our data sets pushed out toward about 1 million neurons. First, we do pre-processing. We then perform some sort of segmentation. It would be a purely morphological segmentation, as it is in the example I'm showing here, generating ROIs that we hope, and in most cases do, correspond to individual neurons. More and more, we're moving to approaches that incorporate fluorescence information into the segmentation process. Currently, our favorite tool is Suite 2P from Carson Stringer at Genelia. From each of those ROIs, then, we can extract an average signal through time and see how that our interpretation is see how that neuron is responding as stimuli are presented. For a very small data set like this, the result might look something like this raster plot here, where we have 400 neurons being represented. These are their responses through time, their delta F over F responses through time. And this represents a series of stimuli that I won't detail for this talk right now. But the next job we have is to say for each neuron, what is its relationship with these different stimuli? What stimulus or what types of stimuli is this neuron active during? What's its relationship with the sensory world? So this is a fundamental type of data that we get out, the information of delta F over F through time registered against stimuli. How do we then try to make sense of what's going on across the brain? So I'm going to talk about a clustering approach we take. This is one of several analytical approaches, but it's probably the one we do first and at least use as a means of getting a sense of what's happening broadly across the brain before we get into details. And what I'm showing here is a raster plot from a very large number of neurons responding to a stimulus train where there are 18 stimuli played across, and these are fictive vestibular stimuli, but that doesn't really matter for right now, from strong stimuli here to weak stimuli here at 18 different times. And we have in green here a cell that is responsive to these stimuli, and in purple here a cell that is probably not involved in processing these stimuli. Obviously, the majority of the cells in the brain fall into this category for any given stimulus. So our goal is to find the cells that have sometimes weak but consistent relationships with the stimuli, and to ignore events like this one. The animal probably twitched here. So we got a big artifact, a bunch of delta F over F that wasn't actually physiological neural activity, and we'll see it in virtually every cell in the brain. So the first thing we do is we extract single traces from single cells across the brain. And then we do a linear regression back to these 18 stimuli. And we find cells with strong relationships, strong regressions to this set of regressors here. We set a threshold, keep analyzing cells that have this relationship and presumably are involved in processing or downstream of processing this sensory stimulus, and ignoring those that don't. Among those that pass the test, we then do k-means clustering. So this is an unsupervised approach whereby we try to identify, put into functional categories or clusters, those cells similar to one another. The goal being that the cells within a cluster will all have commonalities and very high correlations with one another, and cells in separate clusters will be more different from that, but similar to one another in that cluster. We can then take the clusters that fall out of that analysis and regress again to the stimuli, but now with a higher threshold, such that clusters like these, so clear relationships with repeated stimuli, are maintained, and these are the average responses across all the cells in that cluster. A cluster here of cells that's inhibited by this stimulus, and we can take other clusters that come out of the k-means approach, but that are probably produced by, say, artifacts or a rise in signal through time that has nothing to do with the sensory system. They fall below a cutoff value for this regression and are discarded. What this leaves us with then is a set of clusters, each containing usually hundreds or thousands or tens of thousands of cells that have particular response characteristics. And where that almost inevitably leads us is to asking where they are in the brain. Answering that question requires registration of our data, animals to one another, and animals to an agreed upon reference for the zebrafish brain. So that process looks something like this, where for each animal, we take its whole brain. This is the GCAMP signal across the whole brain. We're now ignoring the activity and simply looking at this anatomically. We go through a rather computationally intense process of taking all the animals, might be a dozen animals in a data set, and warping them to one another to provide a reference brain, a registered brain representing all 14, all 12 animals from the data set. Then we register that to an accepted atlas of the zebrafish brain. This has the advantages of first of all, taking a very meticulously assembled template and putting our data into it. And this is also annotated with, I think it's 294 different structures in the brain. So if we see a particular pattern of activity in one of those animals, its XYZ position registers to an XYZ position in our registered brain, and that then registers to the atlas, which tells us that's in the left habenula. And then we know something more about the processing. The result of this is a template larva that has both Z-brain and all the information from all the larvae put in it. And we can drop that into a universal shell, basically just for display purposes. So what might this actually look like? What might the product of clustering followed by registering in this way look like? And I'm going to use a recently published example to show you what it looks like. This is an experiment in which we were interested in habituation, so the process by which larvae ignore stimuli that occur repeatedly without consequence. And so these animals saw 3 blocks of 10 threatening visual looms. So you can see here 3 blocks of 10, and in many cases and behaviorally, the animals sort of quit responding to this after a period of time. So maybe during these first 10 stimuli, there's a reduction in the responses. There's a 5-minute break there, something of a recovery, another reduction. And when we performed clustering, we found that there were different categories of neurons, some which habituated very strongly, responded once or twice, but really never again. Some that habituated moderately. This was more or less in line with the animal's behavior. Some that habituated weakly or not at all. We found neurons that we weren't necessarily expecting to find that were actually inhibited during the course of this visual stimulation in a way that wasn't particularly habituated. And of course, unexpectedly, there were neurons that were responsive in each trial when the animal actually produced an escape response. So clustering gave us these categories, and then the registration process allows us to put that back in the brain and get our first glimpse of the habituating brain. You can see that here, where these different categories of neurons are then put here. For each of these points in space, we have an address, we have a brain region it's in, we have a response profile, and it's color-coded to a cluster, giving us a sense of what that response profile would be. So it's on this basis that we can start doing simple analyses of how information might flow through this network. So those are our microscopes, and those are our analytical approaches. And we've used them to analyze a number of different sensory modalities, each time customizing the stimulus delivery to what we want to study. We've studied the lateral line system. This is a system on the animal's surface with hair cells that bend when water flows. Is very important information to a free swimming fish. So we developed a microfluidics device where we could perform calcium imaging in the brain of this animal while we did controlled pulses of water across its body. We've studied the vestibular system in a way that I'm not going to talk about yet, because that's the one biological story I'm going to talk about in the next few slides. And then either using a speaker coupled directly to the cover slip side of the chamber, or using a monitor outside the chamber, we can present acoustic or visual stimuli to animals and study the brain-wide networks that process those stimuli. I'm going to talk about the vestibular system because this has a nice little physics hook. Much of this was published a few years back, so I've mostly stopped talking about it. But I think it's the right story for this audience because it takes advantage of the strange properties of our microscopes and because it does involve optical biophysics. There's a problem with understanding the vestibular system, and that is that what it responds to is acceleration or a change of relationship with gravity. So your vestibular system becomes active when you move in some direction, when you accelerate in a line, or if your body rolls, if your body changes its relationship with gravity, specifically if your head does. These kinds of movements are incompatible with most ways of detecting brain activity. So you can't spin somebody in an fMRI. You can't accelerate something while you're performing calcium imaging on it. And as a simple consequence of this problem, we understand vestibular processing rather poorly. So the key would be, you'd love to be able to stimulate the vestibular system in a stationary animal and thereby observe what's going on in the brain. And there are other ways to trick the brain into thinking the vestibular events are going on. But in our context, we had to get back to the organs that provide the vestibular sense to larval fish. These are otoliths. Each larval zebrafish will have 2 on each side. And the dogma, which is somewhat true, is that there's one pair, the utricular otoliths, that are for vestibular perception, and another pair, the saccular otoliths, that are responsible for hearing. I was at some lab meeting about 2015 lamenting the fact that we couldn't present vestibular stimuli and also image the brain. And Idia, the physicist who was there, started asking questions that I didn't see the point of. She was asking, what are they made out of? Well, calcium carbonate. And what's the refractive index? And I didn't know, so we looked it up. What are they shaped like? It's like, well, they're kind of flattened spheres, little disc shaped. And she said, I reckon I can apply forces to them with optical traps. I said, what are optical traps? So Idia explained what optical traps are. And what they are is, if we look at these otoliths here, and there's one modeled here, if you take an object that is at least fairly transparent, that has a higher refractive index than its surroundings, and you put a beam, usually an infrared beam, through it, that beam will be refracted. And with the geometry of a sphere or something like a sphere, that refraction, if it goes through the outside edge, will be toward the center. So photons don't have mass, but they do have momentum. And this shift of momentum from directly vertical to slanted involves the shift in momentum of the photons to the left. And Newton then dictates that an equal and opposite force be placed on the object that is producing that refraction, the otolith in this case. So by putting a beam through the otolith, she was actually able to apply a force to this otolith in this direction, in the rightward direction. So the first thing that Idia did for this project was she did a painstaking analysis of the otolith's structure in terms of the refraction that it could put onto objects, that it could put onto beams being focused on various parts of it. Then she dissected out an otolith, and on some fateful day several years ago, applied an optical trap to it and discovered that she could indeed drag this otolith around a cover slip using this laser. So the otolith is trappable. The otolith is producing physical forces, and so that's very useful for us. The question then was, can she apply these forces in vivo in a way that will actually register with and make sense in the animal's vestibular system, which was designed for very different forces potentially. So the next thing she did was she took an animal, embedded its head in agarose, and focused an optical trapping beam on one of its otoliths. And amazing, it was basically the first time she tried it, this worked. This was thrilling. She found that if she applied a force to this otolith, so she's taking this otolith and basically tugging it outward, she found a couple things happened. One thing you can see pretty clearly, the animal's tail bent in a consistent fashion. And if you look very closely, I haven't shown a close-up, but the eyes actually roll a bit too. And this is interesting because these are exactly the behaviors you would expect from an animal if its head is here, like facing toward you, the way I'm looking at the camera. If its head rolled like that, it would do two things. It would bend its tail to try to compensate and stay level. And because the body's rolling this way, it would roll its eyes within their sockets to compensate and keep the visual world steady as the head rolled. Our eyes do the same thing within our heads. So this was really encouraging, both because we saw stuff happening and because, to a first approximation, it's exactly the kind of stuff we'd hope to see. She further described this behavior by either providing an outward force on one otolith or an inward force on the other otolith. Of course, they're being pulled in the same physical direction, which means outward for one and inward for the other if the head were to roll or if it were to be accelerated to this animal's left. And because she was using two optical traps that she could shutter, she can do something you can't do in nature. She could apply this force just to one otolith, but not the other, or the reverse, or with both traps, she could apply them both at once. And she found this initial experiment, it's the one you've seen on the previous movie. If she tugged this otolith out, she saw in red here a bend of the tail that then drifted apparently passively back to center. She pulled this one in, blue, nothing happened during the trap. But when she released the trap, she saw a twitch back in the opposite direction, drift back to center. And only when she provided the entire fictive stimulus, and this is what the animal presumably would experience with both otoliths involved, did she see an active bend of the tail and an active return to the center at the end of that. So that was very cool from a behavioral standpoint. She could then take these empty filter cubes in the microscope, and while producing illumination planes and a detection column here to do the calcium imaging, she could also pipe in her infrared laser, her optical trap, gimbal mounted mirrors to steer each of two traps and perform traps while also performing calcium imaging. And it's through this approach that she was able to define, and those who are paying close attention will recognize these as the clusters that I talked about when I talked about the method. She found a category of cells that were strongly responsive to strong stimuli and also down to weak stimuli. So somewhat graded, but still quite heavily responsive even to very subtle stimuli. So highly sensitive vestibular cells. She found another category that were responsive, similarly much less sensitive and essentially unresponsive to weak stimuli. And she found a category of neurons that were actually inhibited when these stimuli took place. Through the registration approaches that I've talked about, she could then go and give us a picture of the entire vestibular brain. So this is all the neurons she was able to detect that met the thresholds for being vestibularly responsive, and these are their positions within the brain. Once we got these data, first we want to do just some sanity checks to make sure that the things we're seeing here are actually the products of the sensory biology and the sensory motor transition out to that tail bend and those eye rolls. And so we looked at a structure called the nuke MLF. It's a premotor structure controlling movements of the tail. And this was really encouraging because in an event that caused a tail bend to the left, we saw a lot of activity here in the left nuke MLF. We saw very little activity in the right nuke MLF. And in fact, insofar as we saw activity in the right one, this is the side away from the tail bend, some of those neurons were actually inhibited. Those are the purple examples here, the green ones being the excited ones. And then we saw events we wouldn't have predicted. Like here's the cerebellum, because this is all now registered to an atlas. We can pull out just the cerebellar cells and look at them, pull out just the nuke MLF cells, look at them. Cerebellum here. And what we found in this case is we saw lots of vestibular responsive cells, but we saw a concentration of inhibited cells here near the midline, as you can see as it rotates, also sort of on the dorsal side of the cerebellum. Based on the position, these may be Purkinje cells, so inhibition of the Purkinje cells would lead to a disinhibition of cerebellar output. And so this is, although based on these data, we can't really hang any details on this. This is consistent with the idea that there's a cerebellar output, perhaps an error signal related to an unexpected vestibular stimulus, or conceivably related to a mismatch between the eyes and the lateral line that aren't detecting a rolling motion with the vestibular system that is. So this would be a departure point for further exploration of this circuitry, knowing what we can expect to find and where in the brain. And so to summarize what I've shown you so far about our methods and where they stand at this point, I've shown you that we can image the brain, we can see individual neurons through most of the depth of the brain, and we can see activity in each of those neurons. So if I've done my job well, you've seen hopefully some of the advantages of that approach. And if I've done my job too well, you may be believing that this is, or I believe this to be some sort of panacea. And now this issue that I discussed about not being able to address circuits and networks has been solved. And that's very far from the truth. And this is not a panacea. And that's true for a number of reasons. Some of them are strictly technical. So although we can see what's going on in the ventral, the deep part of the brain, our imaging is not as clear as it is in the dorsal brain. Calcium indicators are slow and relatively insensitive, especially when you compare them, say, to electrophysiology. So they're not good at reporting subthreshold activity. Their kinetics are on the orders of, large portions of seconds, or even stretching out into multiple seconds. Mostly this is restricted to immobilized preparations. I've talked about Idia's clever trick for getting around that with the vestibular system. And there are people who design microscopes that can either chase a fish around while doing calcium imaging, or a microscope that can rock. The whole microscope rocks with the fish inside to see what's going on in the vestibular system. So, but for the most part, this is restricted to immobilized preparations. So these are technical obstacles, and I have no doubt that they will fall in the coming years. And then there's some fundamental problems. Well, I shouldn't say problems, I should limitations to this approach. When you see all these blinking points in space, you don't know what they're doing. What neurotransmitter do they use? Are these excitatory or inhibitory neurons? I've addressed whether they're excited or inhibited, but that's different from whether they're excitatory or inhibitory. I'm not going to talk about this today. It's theoretically very straightforward and technically somewhat more complicated to do calcium imaging in an animal, then fix it and perform immunohistochemistry, say, to identify GABAergic cells, and then register your calcium imaging data to your IHC data in order to say, okay, these patterns we observed were happening in inhibitory cells, these patterns we observed were all in dopaminergic neurons, that kind of thing. So that's actually technically hard to do. We're working on it, but I didn't include it in today's talk. Another problem, if the goal is to describe information flow, is this doesn't report on cell morphology. So our GCamp is in nuclei. We do that so we can segment them, but it means that we don't know what their structures are. And we especially don't know how they connect to one another, either structurally or functionally. So we're taking a number of steps to try to address these issues. And the first one I'll talk about is with cell morphology. Here you'll see a big drop in the fit and finish because a lot of these movies have just been added last night. A lot of these data have just been collected in the last week or two. This is an approach we have to identify cell morphology. So this is an animal that expresses a photoactivatable GFP. So with a pulse of UV light, this photoactivatable GFP, which at baseline is quite dark, becomes efficiently fluorescent, and you can see the cell in it. In a densely packed area of cell bodies, which is most of what the zebrafish brain is, we have the goal of using a spatial light modulator to create a very small holographic, as nearly as we can, a point of UV light, so that we can illuminate and therefore label individual neurons, even though all the neurons in the brain are expressing PAGFP. And you can see what is really our very first attempt at this. So this is IDEA taking a spatial light modulator and doing a series of holograms in different positions around the brain, ranging from, and this is just finding appropriate parameters, ranging from very weak ones to rather strong ones over here. You can see an expected increase in photoactivation of the GFP. And promisingly, you can see in these areas individual cells, you can see neurites coming off. If we did more careful imaging, we could see these better. At the moment, she's labeling clearly multiple neurons in each case. She didn't make any effort in this case to dial in the coordinates of individual neurons. But the next step, once we have a grasp of how to create these holograms and how to steer them in X, Y, and Z, will be to perform calcium imaging in an animal, do a very quick regression-based approach to find the types of neurons we're interested in. And we'll know them in advance because we will have done lots of experiments and say that there should be neurons that have exactly these responses in this brain region. We'll take a new animal, we'll find those neurons in a targeted fashion, we'll identify their addresses within the brain, and we'll design a hologram to illuminate them, and if at all possible, them alone as we do that. That will then show us at least a coarse morphology for that neuron and allow us through existing atlases to say what cell type it is, how it's connected, and begin to hang some actual anatomical information on what is otherwise just a cloud of blinking neurons that I've shown you throughout the talk. UV light is not the range you would choose for this approach. So we're also taking a 2-photon approach, and we'll be creating 2-photon holograms in order hopefully to restrict the photo activation further and make sure that it's exclusively taking place in the cell that we're targeting. On the topic of moving to 2-photon, we're also looking at moving the whole calcium imaging process into pulse lasers, into infrared light. You can see here data that, not data, just images that IDEA produced a couple weeks ago. We've just received a laser that we had customized for these purposes. And you can see here just going through water with the fluorescent dye, what a 1-photon light sheet looks like. So this is blue light coming in and creating excitation there. You can see that light sheet there. This is the same effect. And this is the equivalent. Anybody who's learned about 2-photon microscope has seen that famous image where there's the cone of blue light, the 1-photon, and the point at which there's the most blue light, with a 1-photon regime next to a 2-photon microscope, that tiny little point of illumination. This is the planar equivalent, where you can see here just where the plane is densest and narrowest is where you get this excitation. And this is the kind of plane we'd then like to be imaging fluorescence from. But because unlike a scanning 2-photon, we're not narrowing down a point, we're narrowing down an entire plane, we need much more energy to get an efficient 2-photon effect. So our first efforts have been along these lines. This is a movie you've already seen. And this is the 1-photon calcium imaging of a single plane in the brain during acoustic stimulus. This is spontaneous activity, one of the first movies that we've been able to create off of our 2-photon light sheet. And there are things to like about this. The scales are different, don't worry about that. The background is lower. The signal to noise appears to be quite a lot better. Actually, the signals are very clear and bright, but there are problems too. The animal's twitching suggests that we're probably heating it up. And we see areas like this where we're seeing what is not neural activity. These are probably waves of calcium as these cells are being damaged or dying. So there may be a number of reasons why we're getting this. 2-photon light sheets have been done, but they've always relied on having the shutter open for a long time. And we don't have that luxury if we're going to be doing volumetric imaging. And so we've gotten a laser that one has a low repetition rate. So instead of the 80 megahertz that you typically get the 2-photon laser, ours is a 1 megahertz repetition rate. That means that for the same average power, each pulse has 80 times the power. We've gotten it with a short pulse duration, around 200 femtoseconds. And what that means is we have a very high peak energy for each pulse as a combination of the low repetition rate and the short pulse duration. We think the issue that we're facing is with the peak energy of this pulse, and everything we did in designing this laser was about maximizing this to maximize the 2-photon effect. But we think maybe we've taken it too far in the sense that these very high energy pulses may be what's causing the damage here. And there's evidence from this. If Idia images at 1 megahertz, she sees this kind of damage with a given average power, let's say about 100 milliwatts in the specimen. If she cuts then the repetition rate to 100 kilohertz, so a tenth as many pulses, each pulse is the same strength, so only a tenth the average power, we still see that damage occurring. And so we think it has to do with the very high energy in each of our pulses, rather than with the overall average power that we have, that we're seeing these problems. So what we're embarking on now is a period of time when we'll be playing with all of these parameters and combining them in different ways to try to find that sweet spot where we get efficient 2-photon effect and efficient calcium imaging in the 2-photon range, but without heating up or damaging the animal. That's very much a work in progress. In fact, that's probably overstating it. It's work we're just embarking on, and work I'd be very happy to discuss with anybody in the audience if you have any questions or ideas. That's where that work stands at this point. I'd just like to wrap up by thanking my lab and by letting people know, I think some of you know already, but the lab is moving. So we'll be moving to the University of Melbourne, Department of Anatomy and Physiology at the beginning of next year. So I'm looking forward to having all you Monash folks just down the road, looking forward to my new colleagues at the University of Melbourne. And with that, I'd just like again to thank the organizers, to thank all of you for attending, and I'll be really happy to answer any questions you have.
Thanks very much, Ethan. What a fantastic talk. Just really nice, coverage from microscopy building to some really beautiful, biological applications and advanced analysis as well. So for everybody in the audience, please feel free to either raise your hand and we'll unmute you to be able to ask a question, or you can put it in the chat and I'm happy to read that out on your behalf. So it looks like we have one raised hand at the moment, Pete Curry.
Hey, Pete.
Hello there. Am I unmuted now?
You are.
I can hear you fine.
So yeah, fantastic talk. I'm really intrigued by the photo conversion in 2-photon, which is sort of like a holy grail in the fish field, which has been with traditional 2-photons very unsuccessful, I'd have to say, at least phenomenologically. Is there a biological reason for that? And why would the light sheet be more efficient than perhaps the traditional 2-photon photo conversion systems?
So to be clear, we're talking about the targeted photo activation that when I talked about
photo activation. Okay. Yeah. Okay. And that from 0 to green, basically, is that what you're saying?
Yeah, exactly.
Yeah.
So in doing that photo activation, we're not relying on the light sheet. The light sheet we'd just be using for doing the calcium imaging across the brain. And in our context, what that would mean is we're using the light sheet to identify those neurons that we're interested in.
Yeah.
The photo activation itself would then be done also with a 2-photon laser, or the data that you saw today were from a 1-photon laser. That's being piped in through one of the filter cubes down through the 20x objective and being delivered to the brain in a similar fashion there. And so that would be delivered in a manner pretty similar to what you would be getting with a conventional scanning 2-photon, where it's taking the scan head and using that to direct the energy to as nearly as it can a point in space.
So no biological limitation or a photofluorophore limitation of doing that, basically, is what you're saying.
None that we're aware of. And there are tricks you can do. This would both be for photo conversion, photo activation, and even just for standard optogenetics with sort of dancing the point around that target cell. If you have resolution to do that, we might try to do that. But I think in the spirit of walking before we run, we just want to get those holograms tight. We want to make sure that we can accurately dial them into 3-dimensional positions within the brain.
So the volume of those holograms you de novo determine, do you? Specifically, you just do a region and you hope that there's a neuron within it. Is that basically?
Well, that's what you saw today, because we're just getting the SLM working and exploring the parameters of how much energy we need and for how long. But the goal in the future will be to do quick calcium imaging and then a very quick regression-based approach to identify target neurons based on their response profiles, based on their relationships with stimuli. So we've gotten fairly good at saying, okay, this is the brain and these are the different response types. And we could say we're really interested in these cells in the forebrain that are only active during acoustic crescendos because we think we think that's a threatening stimulus, and we're interested in the neural network. We then go to a new animal. We do quick, dirty calcium imaging with a very strong expectation for those response types that we'd see in the forebrain. Do a regression-based analysis, say in this animal, we've identified 57 such cells based on the criteria we've laid out and the thresholds we've set. And then we would go in and knowing their XYZ positions, we would customize a hologram in that animal for one or more of those cells.
Amazing.
that would be calculated in real time while the animal's stationary.
Yeah, well, cool. I'd like to come over and watch that.
yeah, well, if and when we're capable of it, you'll be welcome.
Oh, well, it's very exciting to have you in
Melbourne.
Melbourne. Yeah, I'm looking forward to it a lot. Okay.
Thanks,
Thanks,
you, Pete. I appreciate it. Yeah.
Yeah, thanks. I'll reiterate Pete's enthusiasm. I didn't know you were coming down, and yeah, it'll be great to have you in the neighborhood.
Thanks. I look forward to it.
Yeah, I thought one thing that's really so nice in neuroscience research is this kind of, reference brain atlas where you have that anatomical reference, and you can, if you're imaging correctly, you can align your data with that reference. I think that's quite a feat, and what a tool for the for the community. So with the neural networks, so you're able to kind of identify individual neurons or networks that are responding to particular stimuli. Are those, networks kind of occupying the same space in the brain in different animals? Are they kind of, restricted to a particular purpose, or are they kind of being used for a similar type of stimulus, but perhaps in a different, way for different stimuli?
Yeah, those are, the technical question is generally animals look pretty similar to one another. And we have ways of restricting the data that we take seriously and that we report to those events that are consistently happening in every or almost every animal we observe. So that's something that we view as pretty important. So I can give a technical answer to that question. Everything else you said is exactly what we would love to discover. And so some of that we've been able to do, and we've been able to do that mostly in the baseline processing of water flow, vestibular, audiovisual, these modalities. But then fascinating questions come from there. So how does information that is not directly coded in the primary sensory organs or the first synapse into the brain, how is further emergent information created later in the brain? Those are big questions we want to ask. And if I understood your question correctly, we would not be surprised, for instance, to find cells in the telencephalon that encode, the example I gave to Pete, cells that encode the concept of a threat, something that would then maybe modulate a brain state toward one that's more potentiated toward escape behavior. And we'd like to believe that we could see responses in those cells regardless of whether that threat has an acoustic or a visual or a water flow origin. So we would love to find things along those lines. So increasingly, we're moving away from just doing the baseline descriptions of we make this sound, we show this thing, we move water in this way, and these things light up, which is most of what we've done to this point, to looking at sensory integration, to looking at how these complex networks in the individual modalities remain separate and or overlap in the sensory processing. There are points of convergence. There are these mouth neurons that are responsible for really explosive escape responses, and numerous modalities can tap into the mouth neurons and cause that escape. And so that's nice. Like, we know there's things funnel down to there, and we know they start in different places because they're sounds or sights, or they're different types of stimuli. But how do those interactions occur? And if we can elicit an escape from a subthreshold visual stimulus that coincides with a subthreshold acoustic stimulus, but combined they'll give an escape, how is that moderated? So it gets into all sorts of network level questions. The basic workflow I've shown you is a way of saying, okay, what's everything that's happening? What and where, when, but very little about how and why. So a lot of it now goes to neurotransmitter use, the morphology of the neurons, the connectivity of the networks, and the actual nuts and bolts of information flow through. And probably the best tool we have for that in this system will be optogenetic tests of information flow.
Yeah, it's fascinating, and obviously a very powerful system. So if anybody else would like, this is the last opportunity to ask a question, either in the chat or by raising your hand. Otherwise, I might just prompt you a bit, Ethan. So, your move to the 2-photon light sheet is a really nice example of something that just, technically sounds like the best approach, but has practical limitations, especially when you've got, a really functional system in your standard approach.
Right.
I guess it's, how do you approach, optimizing that, and how much benefit do you think you're going to get from putting in the optimization versus, pursuing your questions in the systems that are working? Is that something that you do in parallel?
Yeah, it's a balance we have to strike, and it's something, if you hope the laser arrives and you plug it in and it works the next day. I don't mean the laser worked, but you hope the approach works. But I think anybody who's been in this business for a while knows that's unlikely to happen. So how hard is it, and how much of my very valuable and skilled physicist's time should go into making this work? Those are decisions I and other lab heads just have to try to balance correctly. It helps we have two machines. So we have one machine just cranking away in the 1-photon range while we're really kind of taking apart and playing with the other machine. That helps. And in some cases, it will simply lead to better data. So better depth, better clarity. We may be able to see subtle things we couldn't see before. But a stronger incentive for us, especially for some of our experiments, is that I was all high and mighty about having no off-target stimuli in this chamber while we're doing the imaging. But we've got this blue laser beam going the whole time, and fish see blue really well. So that's a problem. And they seem to habituate to that laser beam pretty well, and they still respond to stimuli on the screen and that kind of thing. But to say the least, this is not something you choose to have happening during a visual experiment. So especially in experiments that we do that involve vision, there's a strong incentive to move to infrared light, which the fish don't see at all, and to leave them in a chamber that is dark and silent, except for those lights and sounds that we choose to add. And we have a suspicion that when we do that, we'll see all sorts of things about the visual system that haven't been there before. And people have done 2-photon light sheet, including the visual system of larval zebrafish, but probably running into some of the same problems that we have, or simply not having enough 2-photon effect from a traditional pulse laser, haven't been able to do volumetric imaging, haven't been able to attain the frame rate that they need to do this approach. I think that's why we're running into issues where other people haven't before. And we can actually get lovely imaging if we go to 200 millisecond frame rates, but then we can't do any of the stuff we do. So we're pushing into that space, and it is, you put your trust in your skilled physicist, and you encourage her, you make sure she's going to be very well rewarded going forward with authorship and that kind of thing. And I guess that's just part of how you try to make these sort of delayed gratification upgrades in the way you do things. So we have the luxury of trying to do that right now, and hopefully it'll work.
Fantastic. All right. Well, that's a great note to end on. Thanks so much for your time, Ethan. Really appreciate it, especially at this very busy time and ahead of your move. And thanks everybody for turning up. If you missed something in the talk, this talk will be posted to YouTube in the near future, and we'll advertise that as well, so you can come back and check on some details.
Thanks.
All right.
And everybody should feel free to contact me if they have any further questions or anything they'd like to discuss. Thanks very much, Alex.
Thanks, Ethan. All right. Bye, everyone.
Take care.